The first antibiotic enters the bloodstream like a weapon designed specifically for you. Within minutes, members of your colony begin to rupture. Others stop growing, their internal machinery frozen before they can make another copy. The infection that once spread freely through the patient's body is collapsing, and the doctors watching from outside believe the medicine is working. They are almost right, because deep inside the dying colony, you are still alive. You are a bacterium inside a seriously ill hospital patient, but the name Superbug does not mean that you are stronger, larger, or more intelligent than ordinary bacteria. It means that the drugs normally used to control bacteria like you can no longer be trusted to work. Resistant infections can become difficult or sometimes impossible to treat, which is why antimicrobial resistance is considered one of the world's most urgent health threats. You were not born with a plan to defeat medicine. Bacteria do not study antibiotics and consciously design defenses. Your advantage began with chance. Somewhere in your DNA, a mutation slightly changed one of the structures targeted by the drug. The change may have been useless under normal conditions, but when the antibiotic arrived, that tiny accident became the reason you survived. Around you, the sensitive bacteria disappear. You divide. Your descendants inherit the mutation, and soon the infection is being rebuilt by bacteria the first drug cannot easily kill. The medicine did not teach you how to resist it. It removed the competition and left resistant survivors behind, allowing natural selection to transform a rare trait into the dominant one. The doctors respond by changing antibiotics. The second drug attacks in a different way. Some antibiotics interfere with construction of the bacterial cell wall, others block the ribosomes that build proteins, and others disrupt processes needed to copy or maintain DNA. Every new drug creates another selection test. This time, your protection does not come from your own mutation. It comes from a stranger. Another bacterium drifts close and extends a thin bridge toward you. Through that connection comes a small circular piece of DNA called a plasmid, carrying genes collected from bacteria that survived antibiotics somewhere else. Bacteria can exchange resistance genes through processes such as conjugation, allowing useful defenses to spread between cells and sometimes across species. In moments, you inherit a survival trick that may have taken years to evolve elsewhere. The second antibiotic reaches you and tries to enter, but the new gene gives you an enzyme that can break the medicine apart. If the attacking drug belongs to the beta-lactam family, a beta-lactamase may cut open the chemical ring the drug needs to work. More dangerous versions can destroy antibiotics reserved for severe resistant infections. The drug arrives intact. Your enzyme grabs it. The weapon falls apart before reaching its target. A sample is taken from the patient and spread across laboratory plates containing different antibiotics. The doctors are no longer choosing treatment by habit. They are testing which drugs can still stop you. On the report, several names return with the same result: resistant. A third drug is selected, one your enzyme cannot destroy. This antibiotic must cross your outer defenses before reaching the machinery inside you, but your membrane has changed. Tiny channels called porins normally allow certain molecules to pass through the outer membrane of many gram-negative bacteria. By losing or altering some of those channels, you reduce the amount of medicine that can enter. Limiting drug uptake is one major form of resistance. A few molecules still slip through, but another system is waiting. Embedded in your membrane are efflux pumps, microscopic machines that capture harmful compounds and throw them back outside. The antibiotic enters, the pump catches it, and the drug is expelled before enough can accumulate to kill you. It is like trying to fill a leaking room while powerful pumps continuously empty it. The doctors increase the dose, but the patient's organs can tolerate only so much. Your defenses do not need to make you completely untouchable. They may only need to push the effective dose beyond what can be given safely. Then the next attack begins. This drug reaches its target, but the target is no longer shaped the way the medicine expects. A mutation has altered the binding site, like changing a lock after the enemy has already made the key. The antibiotic cannot attach properly, while the process it was supposed to stop continues. Target modification, drug inactivation, reduced entry, and active efflux are among the central ways bacteria survive treatment. Still, you are not fighting alone. You and thousands of other bacteria attach to a catheter inside the patient and build a biofilm. The colony surrounds itself with a sticky protective matrix, creating a fortress where resistance genes can move between neighbors. Antibiotics may penetrate the outer layers but fail to reach every bacterium at an effective concentration. Cells buried deeper may grow slowly, making drugs that work best against rapidly dividing bacteria less effective. Some bacteria enter an inactive state and wait. They are not necessarily genetically resistant. They are persister cells, temporarily tolerant, because their biological activity has slowed so dramatically that the antibiotic has little to attack. When treatment ends, some wake, divide, and help the infection return. By now, the patient is dangerously ill. You enter the bloodstream, and the body responds with inflammation intended to contain the invasion. If that response becomes dangerously dysregulated, blood pressure can fall, organs may receive too little oxygen, and sepsis can develop. The medical team removes the infected catheter, drains infected fluid, and searches for tissue where you may be hiding. Antibiotics are important, but controlling the physical source of infection can be just as critical. A fortress cannot protect you if the entire surface holding it is removed. Your population falls again. Only a smaller group remains, carrying a patchwork of resistance genes, an enzyme from one ancestor, an altered target from another, reduced porins, powerful efflux pumps, and plasmids that can be copied and shared. No single bacterium planned this combination. It was assembled through mutation, inheritance, gene exchange, and repeated selection. The doctors reach for a last-line antibiotic. This is not necessarily the final antibiotic in existence, but it is one of the few medicines still realistic for this infection. It may be more toxic, less reliable, difficult to use, or carefully reserved because losing it would leave dangerously few choices. The drug enters. For the first time, your defenses begin to fail. Your outer membrane is damaged, your internal balance collapses, and bacteria around you die faster than they can divide. Your shield was never perfect, only layered, and this attack has found a weakness. Then the laboratory notices something troubling. A tiny fraction of the colony is still growing. Perhaps another gene has modified the part of the cell the drug attacks. Perhaps the drug concentration is too low inside damaged tissue. Perhaps the survivor is protected within a biofilm or carries a rare mutation present before treatment began. Whatever the reason, the nightmare repeats. Susceptible bacteria die, survivors reproduce, and the next generation begins with one fewer vulnerability. You survived every drug the doctors tried. That does not make you immortal. A carefully selected combination may overwhelm several defenses at once. The immune system may clear what remains after treatment reduces the population, removing infected devices, draining abscesses, isolating contagious patients, cleaning hands and surfaces, preventing infections, and using antibiotics appropriately can reduce the opportunities that create and spread resistance. Your greatest advantage has never been intelligence. It is reproduction. A human generation takes decades. You can produce descendants rapidly, and every replication creates another chance for mutation. You also live in enormous populations and can exchange useful genes with neighboring bacteria. Repeated antibiotic exposure gives resistant organisms more chances to survive, multiply, and spread. Outside the room, the medical team puts on gloves and gowns. Equipment is disinfected, and the patient is isolated so you cannot ride on a hand, device, or contaminated surface into another vulnerable body. For the first time, your path forward begins to close. You survived the cell wall attacker, the protein blocker, the DNA targeting drug, and the medicine saved for emergencies. Yet the most dangerous part of your story is not that one bacterium became unbeatable. It is that the instructions protecting you can be copied, shared, and carried into infections that have not happened yet. You are not a monster created by a laboratory, and you are not proof that antibiotics have stopped working everywhere. You are biology responding to pressure. One survivor multiplied until an old treatment lost its power. The doctors stare at the resistance report, searching for one remaining weakness. Inside the patient, you divide again. And somewhere in your descendants, another mutation is waiting to be tested.